Dear friends, I'm honored to introduce you Popov V.O, the member of Russian Academy of Sciences, the director of the National Research Center for Basic Biotechnology of Russian Academy of Sciences, and the expert in structural biology and biotechnology. Please.
Thank you for this kind introduction. I'm the first time in Shenzhen, and I'm very impressed by the university, and it's my privilege to tell you some words about our recent research in structure functional studies of macromolecules. And basically, I will be speaking about structural biology. Structural biology is one of the pillars of modern science, modern life sciences. Every second or third paper published in Nature is heavily decorated with fine structures of macromolecules and its complexes. But why? Why do we need structures? There is such a saying, seeing is believing. And indeed, 80% of the information from the outer world we receive from our vision. And when we say the rest, 20, we receive from all our senses. Structural biology is indispensable when you look for new drugs during drug design cycle, when you design new biocatalysts, and of course, you need it to understand how life is organized for all this. Just a moment. Okay. No pointer here, probably. This. Oh, I see. I need a mouse to operate it. Okay, and you need, of course, structural biology to understand the fundamentals of how a cell is organized, all this intricate process which take part inside the cells, is processing, folding, trafficking, signaling, and so on and so forth. And again, the journal Nature gives the following definition of structural biology. Structural biology is the study of the molecular structure and dynamics of biological macromolecules and how these alterations in their structures affect their function. An important thing is the mentioning of function, the interplay of structure and function. So on this slide you see a couple of structures. This is myoglobin and this is hemoglobin. And they have three features in common. First of all, they are transporters of oxygen in our bodies. Secondly, these are the two proteins, the first two proteins, which 3D structure has been determined several decades ago, and they also share the same Nobel Prize. So structure is required to understand why, for example, a hemoglobin can reversibly bind oxygen of CO2, why myoglobin not. It's because of its quaternary structure arrangements, about the allosteric properties of hemoglobin. And so the following dogma emerged that the structure determines function. Like in molecular biology, you have this the DNA, the transcription and translation. But it's not that simple in the case of structure and function. For example, the same protein, such a simple protein like cytochrome C, has about more than 10 different functions in our organism, in different cells. It is, of course, an electron transport protein. It could be a catalyst, and it also be an important trigger of apoptosis in mitochondria. And of course, there are a number of proteins that don't have determined 3D structure at all. That is so-called intrinsically disordered proteins. They acquire definite structure only when they see their partner, yeah? When they form a complex with some other molecule. There is other class of proteins which are called moonlight proteins, if you heard about that. That is the proteins with the same functions, the same structure, but in different compartments of the organisms that perform different functions. For example, glycerol triphosphate dehydrogenase, a very common and popular enzyme. So the situation is much more complex than first expected, and the interplay between structure and function is very complex. So structural biology has accumulated a number of instruments to study structure of macromolecules. That is X-ray, nuclear magnetic resonance, cryo-electron microscopy, small angle X-ray scattering, and different modeling exercises like quantum mechanics, molecular mechanics, talking with artificial intelligence, which we just talked about. All these methods have their advantages and disadvantages. There is not a single method of choice. And the message is that you have to pick up as many methods to attack the concrete problem. We are talking about integrative structural biology. That is the modern trend. And before I turn to structure, I would like to give you some overview how the statistics of structural biology looks like at the moment. So, the technological advances in the last decade of the 20th century resulted in the avalanche of structures. Yeah? So it was an exponential growth, now we have a linear growth of the protein data bank, and each year we require more than 10,000 new structures, and it is already exceeded 200,000. I haven't reviewed it since March, but these are more or less the same figures. And just recently the structural bank was divided into two parts, the experimental one and the forecasted one, as a number of alpha-fold structures have been deposited, and this number is sufficiently, so much more in numbers than the experimentally determined ones. It's important to understand the dynamics, the input of different methods into the statistics of the protein data bank. So the X-ray remains the workhorse of the modern structural biology. It accounts for nearly 90% of all the structures. But recently, and this is the rate how the structures are accumulated by different methods, and the scale is logarithmic, it's not linear. You see that there is a rise, a very big rise in the cryo-electron M structures, and probably soon the cryo-electron microscopy will be in line or even exceed the number of X-ray structures. It's more or less the same figure, yeah, but it shows the technological advances that resulted in this nearly 200,000 experimental structures. Yeah, something wrong with the scale, but here is it. Yeah, here it is. So you see also a constellation of the Nobel prizes that were awarded in the field of structural biology of the past several decades. So it's very rewarding and fruitful to work in this field. You have a real chance to be prominent, to get something real. So, but all these structural projects are very risky ones. So, for example, if you want to obtain a structure with a conventional X-ray method, and if you start with the gene, for example, from a gene bank, you have to clone, to express, to isolate, crystallize, and to obtain structure, and all this stage is subject to failure. Yeah, and the percentage of failures is of course different. The most risky ones are to some extent expression. Crystallization is very ambiguous. It could be only up to 30% successful. And even if you obtain crystals, it doesn't mean that it will diffract properly. You could spend decades, years to obtain proper crystal to gather all the necessary data. So the problem is that, as you know, the probability of the complex event is multiple, the product of the probabilities of the individual ones. So if you multiply all these probabilities, you arrive at these figures. If you are an optimist, you can say that the success rate is about 20%. If you're a pessimist, then the great success rate will be about 3%. Me personally, is close to the pessimistic view. I evaluate the success rate of any structural project is about 5%. It's about the same success rate as in the startups. 95% of the startups fail and only 5% proliferate and further develop. So, what does it mean, this very low success rate? It means that the simpler the protein, the easier to obtain its structure. And the result is that the protein data bank is very redundant. It's 200,000 structures there, but only individual proteins, real individual proteins are about 20-25% of them. There are nearly 1,000 structures of only lysozyme in the protein data bank. So the easier the protein, the easier it is to crystallize, for example, the easier to obtain its structure. When it comes to a really complex protein with prosthetic groups, metal centers, if it is glycosylated, different complexes, you have a problem. A good example is the membrane proteins. They comprise about 25% of all the targets of the big pharma companies. They comprise 25% of the proteome in our body, but only 3% of the net protein data bank, because it's very difficult to work with them. They're not readily crystallized. Probably the new techniques like cryo-EM microscopy will bring some progress into the field. And then, of course, as I mentioned, there are intrinsically disordered proteins and so on. So if you look at the statistics on the right column here, and this statistics comes from the so-called Protein Structure Initiative from the United States, when everything that was isolatable was subjected to crystallization and obtaining structure. So you see that when it comes to simple proteins, your figures are in thousands, but when it comes to really tough things, the numbers are considerably decreased. So the problem stays the same, yeah? And you have the message to take home that you have to tackle the problem with the multiple approaches, that you have to use this integrative structural biology and not to reside on only a single one method. So now, going back to the, not back but forward to the experimental results that were obtained in our laboratory over the past couple of decades. So I'll tell you in brief about several projects that are related to different classes of proteins, simple NAD-dependent dehydrogenases, metalloenzyme, heme-containing proteins, and I'll try to illustrate my thesis that I put forward in the introductory part of my speech. So the first example is from the field of simple NAD-dependent dehydrogenases. It dates back to the 80s years of the past century, At that time we were all very romantic and believed that very soon all the traditional chemical catalysts will be substituted by the biocatalysts and all the chemistry will be green, won't interfere with nature and so on. So, and if you want to, for example, to obtain some final product, for example, some chiral alcohol or chiral amine, you need NADH or NADPH as a reducing agent. And that is a very common task in different organic synthetic routes, especially when you synthesize different syntones. Syntones are intermediates for producing drugs. So you'll have with these reducing agents, NADP or NADPH or NADH, spent at stoichiometric quantities, and they're very expensive. The idea is to regenerate them. So you need an auxiliary enzyme that will make from NAD or NADP again its reduced form. And the enzyme of choice is formaldehyde for several reasons. First, because the product is CO2, it escapes the reaction and it drives the equilibrium of all the process to the completion, to the desired product. So it has, the enzyme is readily available, stable, has a pH, wide pH optimum, high stability, and so on and so forth. So it's very versatile and convenient enzyme to work with, and you have plenty of publications over past decades of different applications of this formaldehyde for different purposes. So very soon we got interested in the structure of this enzyme, and our first publication appeared in 79, when you see there were only few structures in the protein data bank. There were the early stages, and every new structure was happening. It was widely discussed, it was something very special in the field. So very soon, quite soon, in several years, we obtained crystals of this formaldehyde and came up with a 5-angstrom resolution. At that time, it was not a bad thing. And because there was no molecular graphics at that time, the models of the proteins were cast either in plastic, like this one, or made of fibers and balls. So very, very primitive. But you see already when we came with our first structure, the number of protein structures increased to several hundreds. Yeah. So when we came with 3 angstrom, the numbers and structures were already close to 1,000. And when finally we got a nice picture, when we saw everything in detail about the active site, there were already 3,000 structures in protein data bank. So in a few years, it increased from several dozens to several thousand, and while we were doing our work. Okay, and finally, when we over some time got to nearly atomic resolutions, they were already in tens. So it took, you see, several years, from more than 10 years, to accomplish this route, and now it is done very easily. Yeah, there are special programs, special software packages that you can just feed your raw data into this software package, and raw data, that's what you obtain from the X-ray machine, for example, from synchrotron hybridization source. And in about 3 hours, you obtain a very nice structure. So 3 hours nowadays, and 10 years of research in the 80s of last century. That's what technology does with science. Yeah? And if, for example, 30, 40 years ago, each structure was happening, as I told you, now it's work for a student of the second, third form of education. Yeah? And so it's really the difference between the science in former days and its technology nowadays. Yeah. A lot of people do, for example, comparison of nucleotide or protein sequencing, do blasting. They don't understand the algorithm that underlines this. Yeah? And you don't need to actually, because there are people who devise this. The same with these software packages with the structure determination. You just press a button and the machine does for you all the rest. And the majority, absolute majority of people, they don't understand what actual machine is doing. Yeah? And my advice to the students, just try to learn to understand what the algorithm is really doing. It's about artificial intelligence and blah, 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 before relying upon it totally. Anyway, so this formaldehyde-hydrogenase is the simplest and most versatile model for energy-dependent dehydrogenation, because it's one of the simplest reactions. Only one CH bond is broken and one new CH bond is built. So we managed to trace all the conformational changes in protein structure during the catalytic turnover. We so formulated the molecular mechanism of action because we understand how the active site of the enzyme is organized in very fine detail. We verified it by, of course, mutagenesis and with these theoretical studies. And when we arrived at very high near-atomic resolution, we were happy that our structures completely verified the proposed theoretical mechanism of this dehydrogenation. At this resolution, you can see very minute changes in structure. For example, in the bond length of this between the aromatic ring and this carbon. Yeah? And what does the enzyme when it binds the cofactor? It rotates this group and takes it out of the plane of the aromatic ring that results in build-up of the positive charge of this C4 position of nucleotide. It becomes more electrophilic and easily is attacked by this hydride ion, which is transferred from the formate, which is the substrate. So just about 15 years of work and a very detailed mechanism that is now in a lot of the textbooks. Yeah. Another example is also about very simple class of proteins. These so-called zinc-associated domains that about the architectural proteins of eukaryotes. Our chromatin is very complexly organized, as you know, and to express it you need structural reorganization, and there is a special machinery that activates chromatin, and there are a number of proteins which shape it in 3 dimensions. They are called architectural, and some of them, they bring different parts of DNA closer together, forming these topologically associated domain stats where genes are expressed in the coherent way. So architecture of proteins determine long-distance interaction within the genome, and usually they have a very complex structure. They have this part which is called zinc fingers domain. Zinc fingers are special structures that are devised by nature to interact with nucleotides, RNAs or DNAs. And they have some other domains which determine the interaction with their counterparts. So to crystallize, for example, such a big thing is very problematic because these zinc fingers usually don't crystallize at all. So it's done in portions. You dissect the big molecule into the domains and do them separately. So these zinc-associated domains, they're very simple. they're composed of two alpha helices, a very simple beta sheet, and if you have this zinc ion here complexed by 4 cysteine residues, and proteins containing these Z domains are quite numerous. For example, in arthropods, and they possess several functions. They can be, for example, activating, silencing, and so on. Yeah? They have very low homology, and their specific properties is they form dimers. So just acting like a clip, so at Z domains on different parts of the chromatin, they put them together, forming these topologically associated domains. So it's a very simple structure, and the very simple question: why they homodimerize? Why you don't very rarely observe heterodimerization? Why they are so specific? Yeah. And we solved a number of these structures. They are all very similar. Yeah, I told you about these alpha helices. There are different bits that could be auxiliaries. They're not so special for their function, but this structure, a very simple one, can accommodate a lot of primary structure. So it's very a lot of amino acid residues can appear within these two alpha helices. Yeah. It's very tolerant to different amino acid substitution. And the monomers, if you compare them, are very much alike, but if you superimpose two different one over each other, you will see that they diverge, because this deviation in their primary structure is this one alpha helices. And that is the reason, that is the explanation why they homodimerize and don't form heterodimers. And it's very important for the evolution of the organism, because after the gene duplication, you need a platform that can rapidly evolve to gain specificity after this gene duplication. That's just an example of a very simple structure and this high functional versatility. Yeah. Another example comes from the area of extremophiles. As we are higher organs, the eukaryotes, we live, our upper limit of survival is about 55 degrees. I have been told that in summer there is 40 degrees here, probably a bit more. So we are very close to this survival limit. While the prokaryotes, they can live well up 100 degrees centigrade. The upper limit, documented limit of life nowadays is about 120 degrees. And you should be very, very special to live under such a very harsh conditions to perform, not only to be stable, but to perform some special structure. So we decided that it's interesting to understand the structural basis for extremophilicity, and there was also a practical, so some interest to obtain new enzymes stable at severe conditions for some practical application. I'll provide you two examples, and one of them is short-chain alcohol dehydrogenase. It's one of the most stable proteins, enzymes ever described. Probably it shares, it is within the first 5, definitely. You can boil it for 1 hour. It withstands the temperature. It melts only at 105 degrees centigrade. It likes high salt, 4 molar sodium chloride. It is stable in 8 molar urea, and which is very important. It's also stable in organic solvents. So this properties of polyextremophilicity are very interesting, how one protein can accommodate all these things to be stable in organic solvent at high temperature in 8 molyuria. This all quite different mechanism of enzyme unfolding, protein unfolding, and inactivation. So the clues should be somewhere in the structure. But when you superimpose the structure of this very stable alcohol dehydrogenase from these organisms onto the mesophile or thermophile structure, you see no difference. The structures are more or less the same. So there should be something in very fine details. And when you look into the structure of this protein, you understand what's the reason. So it is covered by, the surface of this protein is covered by network of electrostatic interactions and salt bridges. It has the highest percentage of the salt bridges, not only among, of course, mesophiles, but extremophiles as well. You see, these electrostatic interactions, they result in this network of salt bridges. And you can think of this protein as an ancient night covered in armor, yeah? And all these hot molecules that attack this protein from the bulk, they are repelled easily by this network of electrostatic interactions. And moreover, some of these interactions, they disappear under ambient temperature and appear only at high temperatures. So, as you see, it's quite so active at 80 degrees, that's the optimal gross temperature for this organism. But when you cool the reaction mixture, the reaction slows down and nearly goes to zero because it becomes very rigid. And to perform catalysis, you need to be very flexible, yeah. And because this molecule is tuned to survive at high temperatures, it's only flexible at these increased temperatures and completely rigid in the ambient. And that is the proof of a versatile, universal principle of enzyme catalysis, that the enzyme is tuned to perform its optimal activity under the optimal of the gross temperature of its organism. And another example comes from the world of the multi-heme enzymes. So as I told you already about I mentioned about hemoglobin and myoglobin, that is hemoglobin and multi-heme proteins play a very important role in our organism. For example, cytochrome P450, it is the detoxifying agent in our liver. Catalase, one of the most active enzymes in nature. Its catalytic turnover is close to the diffusion limit. Cytochrome oxidase, it's a part of the oxidative reductive chain. Already mentioned about apoptosis signaling and these multi-heme proteins. They are, of course, electron carriers inside the cell, as well as they transfer electrons from outside the cell to external carriers. And we came into the field quite all of a sudden. We were studying this extremophile organism, and there was one microbiologist. We have an institute of microbiology incorporated in our research center, who told us that he has an organism that has an enzyme with very unusual properties. Probably it is dependent on such special metals like tungsten, molybdenum, and vanadium. So we decided it's very intriguing, decided to isolate it, and we came up with this nitrate reductase, which contained 48 hemes, that is composed of 6 identical subunits. And once upon a time, it was the enzyme which accommodated the biggest amount of hemes. We were the record holders. And when you inspect how heme are so arranged within the proteins, they always have the same topological arrangements, the same scaffold. Never mind, there are 8, 10, 5 or so many. So these are 8 hemes, for example, in the subunit of whatever, either this or that or this one enzymes. They're always arranged in one and the same conservative way. I don't have time to discuss it in details, but it allows the efficient electron exchange and transfer of electrons to the distances of several, sometimes dozens of angstroms or even hundreds. Yeah, so it's quite a long distance with electronic conductivity. So we were happy with this enzyme. We were very much impressed with it, but then several years ago they emerged another monster. It contains nearly 200, you see, these hemes in one molecule. It's also, again, this conservative, so scaffold template of 8 hemes. It's arranged in 3 identical subunits, and that is octamer of these functional trimers. So imagine such a protein stuffed with these 200 hemes that is like a capacitor flowing somewhere in the cell. It's really very impressive. Even these 48 hemes are quite a lot, but nearly 200 is it's probably not a limit. And so when we so these enzymes of the heme nitrate reductase, it's really a nanomachine catalyzing these very complex reactions. 6 electrons and 8 protons are transferred during catalytic cycle, and the substrate doesn't leave the enzyme active center. It is transferred either to ammonia or hydrogen sulfide. That is 2 of the 3 most complex reactions in biology. Another one, 6 electron reaction, is the fixation of nitrogen catalyzed by nitrogenase. So it's very interesting to understand how it's all happening in this enzyme active site. And actually it's very, for the enzymologist, it's very intriguing and very beautiful. So it has a very long this substrate channel, which is positively charged at the entrance to attract the negatively charged substrate. And it's positively charged, negatively charged, at the outlet to expel the positively charged product. And the substrate is expelled not to the bulk, not to the outer, but inside the protein, into this big cavity between the subunits through these negatively charged nozzles. And you see this, the constellation of these hemes, yeah? I'll miss it, okay? And the active site has a very complicated structure. You see that there is a special covalent bond between this tyrosine, which participates in the reaction donating these protons, and the adjacent cysteine residue. It results in the change of its pK. It becomes more acidic and facilitates the catalysis. So it's really a nanomachine with this dimension. It's 140 angstrom on the longest axis, with a lot of void volumes inside. Again, another example about these multi-heme proteins. It's about how to deliver electrons from external compartments of the cells to the outer space. And you know that there are 2 kinds of cell walls, gram-positive and gram-negative microorganisms. Yes, they differ in the arrangement. In gram-negative, you have a lipid bilayer and another outer layer with the periplasm in between. And for gram-positive, you have this periplasmic space, but you have thick, very thick proteoglycan layer. And sometimes it's covered by a special layer of proteins which is called S layer. So the electrons have to penetrate all these obstacles to reach the external electron acceptor. Yeah? And it was well studied over the past several years with the electronegative microorganism. So it is composed of 3 proteins, one of them 2 multi-heme proteins, so this containing each 2 hemes that are put inside this another protein which forms this barrel-shaped structure. And it was considered that probably the same could apply for gram-positives, but unfortunately we have a young postdoc in our institute who didn't like this idea, and he showed that in gram-positives quite another multi-heme proteins are expressed when they grow on the external electron acceptor, is soluble electron acceptor that is a mineral of iron. And one of them he called outer membrane multi-heme cytochrome, which contains 11 these heme groups. And it turned to be a very elegant and nice structure. It comprises 3 functional modules. So the 11 hemes are incorporated within the N-terminal domain, and another domain which is devoid of hemes is used for anchoring to the S layer. It's S-layer binding domain because it has very similar structure to the S-layer proteins. It has a positive overall charge, which facilitates its incorporation into the negatively charged proteoglycan layer. And the electrons are injected into these subdomains that comprises two hemes, hemes 1 and 2, and they are transferred via the chain of another 9 hemes. Either they are transferred to soluble electrons acceptors like phenolphthalein that work like shuttles, transferred it ultimately to insoluble electron acceptors, which is iron insoluble compound, or it can so directly interact with the surface of the mineral, and because we have a very detailed structure, we were able to locate all the sites where the catalysis takes place, or through which the electrons are channeled into the external substrates. Probably one more example. As I told you, we have a lot of structures already accumulated, hundreds of thousands, while the functions, many of them are obscure. In the protein data banks, there are a lot of annotations that say that the product of heme, product of gene, whatever, putative, for example, hydrolase. So a lot of structures don't have allocated function. And the illustration for that is the number of enzymes we know we have discovered over decades. Remember this curve with growth of data bank, protein data bank, hundreds of structures, hundreds of thousands of structures. Well, up till now we know not more than 8,000 of different enzymes. And only several years ago the number was quite low. It increased only in the past several years. And the number of enzymes could be the depiction of the number of functions, correlates with the number of functions we know more or less. So to discover a novel enzyme is not an easy thing. And again, you need to know where to look for it. And the main source of biological diversity is microbial diversity. You need to have good microbiologists in the team if you look for that. And we had this Dmitry Sorokin, who said that one of his bugs, microorganism he was working with, has very special properties. It can grow on cyanide as a single energy and carbon source. The growth is copper dependent, and the products are elemental sulfur. And these structures inside the cells are the incorporated sulfur inside the cell. So you can draw the reaction, the equation of the reaction, basing on this simple assumption. And it was with this very detailed information about the putative enzyme, it was very easy after some biochemical manipulations to locate it in the cell, to isolate, and we were able to characterize it structurally. And we were extremely lucky, as if we pulled out the joker from the deck of cards. It was not just a novel enzyme, but it catalyzed the reaction novel to the chemistry of cyanide. But what is more important, we discovered a novel copper cluster. The copper-dependent enzymes are not so numerous. There are about 20 of them, and about 7 types of copper active sites are described for this class of enzymes. So we were lucky that this arrangement of 3 copper ions, they form quite new copper sites, that were not described previously. And because we had very detailed structural information, we were able to understand the mechanism of this reaction in very fine details. We can see how the substrate is bound inside the active site, how it is positioned by coordinating with these 3 coppers, how these coppers, you see one of the coppers moves during the catalysis, how the attacking water is activated by the adjacent amino acid residues, and we were able to verify it by quantum mechanical calculations. And then we were also able to isolate and get a structure now with NMR of the cytochrome, that is the gene of coding for which is located close to this thiocyanide dehydrogenase. So again, we are trying to apply several methods of structural biology to tackle the problem. So we were able to locate the regions through which this acceptor interacts with the enzymes, and it was easy to trace the electron transport chain leading from this one of the copper ions to the heme of the accepting cytochrome. And we applied there is a special body, international body, that verifies that you discovered something novel in the field of enzymology. So you know this enzyme classification, so there is a special commission. So we applied for that we discovered something new, and we were granted this new enzyme, which they called cyanide desulfurase. So altogether, we, I think, described more than 150 structures in the past 20-30 years, but we managed to discover only one new function, that is new enzyme. So that is the ratio between the function and structures nowadays. Structures are easy, functions are difficult and rare, novel functions. And that's where this interface are the most interesting thing to happen in the future. Okay, the last example showing that how you should apply different approaches to solve a problem, this integrative structural biology approach. That is a butyryl cholinesterase. You know a lot of probably acetyl cholinesterase. That is enzyme that determines how the nerve signal is transported in our organism. And acetylcholinesterase is subject to inhibition by different nerve agents, yeah, toxins, yeah, that are warfare agents. And butyrylcholinesterase it stoichiometrically binds this toxin just taking away this toxin and protecting acetylcholinesterase. It is in service with the US Army in the White House as an antidote. So the structure of it is a tetramer, and it's highly glycosylated, and because it is highly glycosylated, it's not crystallizable at all. And for more than 15 years, a lot of people tried to do that with very little success. Of course, they obtained a lot of structures of individual subunits that were devoid of, very deglycosylated, but it was of no help. And the model was suggested that looked like a garlic with 4 subunits and this 4 bundle of 4 helices gluing them together. Yeah? And our idea was to, at that time we were just beginning our exercises with cryo-electron microscopy, and we decided to combine different techniques. So we were provided by this sample of this butyrylcholinesterase by our old friend Patrick Masson. It's not all of a sudden that he is in a military uniform that just underlines, emphasizes the interest of which people this type of enzyme usually attracts. And so he provided us with a sample of the protein. Very soon we got the first cryo-electron maps. They were of very poor quality, but it was not so important because we could easily incorporate the already available structures available at high resolution into this envelope we obtained. And just immediately we understood that the model that was suggested wasn't correct. Our structures looked quite different. So this bundle of helices was not protruding, but it was hidden inside the cell. So the next exercise was to model, as I told you, the enzyme is very glycosylated, so it was important to understand how it affects the protein properties. And glycosylation increases the radius of saturation by about 50 angstroms. And so we modeled, we incorporated all these residues and made the molecular dynamics so modeling. And it's interesting to find out, for example, if you look closely at this rotating molecule, you will see these yellow patches. Yeah, these yellow patches, they color the vicinity of the enzyme active sites. For example, here and here. So the active sites of this enzyme, they open on different directions. This face forward and this one face backwards. It was also quite unexpected from the previous model. And the final modeling was verified by small angle X-ray scattering. So a combination of 3 methods, X-ray crystallography that was done by somebody else, previously cryo-electron microscopy, molecular modeling, and small angle X-ray scattering resulted in the very nice structures that completely coincided with also experimental data. So I'm very close to the end. A couple of more slides. So what are the perspectives of structural biology? I nearly didn't touch some topics like cryo-electron microscopy. That is a rapidly evolving field in the area, and several years ago there was a paper paper in Nature with the title "The Future of Structural Biology Will Not Be Crystallized." So it was about the progress of electron microscopy. So, of course, membrane proteins, which I already told you, comprise the majority, nearly 50%, I think, of the potential targets for the big pharma. I haven't mentioned anything about so-called X-cell, that is a synchrotron radiation source on free electron laser. That very sophisticated machine, extremely expensive. It allows you to obtain, to perform time resolved experiments and to make the movie, the molecular movie. You can initiate the reaction by femtosecond pulses and detect the structural changes that accompanies the turnover of the catalytic process. And very so intriguing and very fastly evolving area of structural biology research. And we already discussed here the interplay between the science and artificial intelligence. Artificial intelligence is the slang, it's the algorithms of deep machine learning, to be more precise. And here is a picture from one of the recent papers from Nature, which shows how rapidly this approach is picked up by the scientific community. In the December of 2021, the first publications of the open code of AlphaFold appeared, and you see how fastly it was acquired, how many papers appeared in the past only 2 years, yeah, and many more to come. But of course, this AlphaFold is not panacea. It's not. It has some limitations. It has problems with prediction of the structure of different complexes, with small molecules, with prosthetic groups, and so forth. But the area develops so rapidly that probably they will be overcome already in several years. So what is the take-home message? The take-home message is about integrative or complex structural biology approach. You need not to reside on a single method, but to try to combine them depending on the object you are studying. It could be crystallography, nuclear magnetic resonance, cryo-EM, small angle, quantum mechanical, molecular mechanical, of course, this artificial intelligence. So there is such a Russian saying that if you run after two hares, you will catch neither. And that is this sketch showing a user who wants to get some information about the structure of his object, his molecule, and a number of approaches he can use. So the more hairs you go for, the more approaches you can harness, the better for your results, the closer you will be to the success. Remember my figures about the success of the structural biology project, several percent. You have to increase it. Yeah, you'll have to try to increase it as much as possible and to try as many approaches as possible at the moment. And again, about the artificial intelligence, probably you've heard about the man whose name is Fukuyama. He is American sociologist and economist, and in the beginning of the century he wrote a famous essay, The End of History, that was not about life science, but was about sociology. After the end of the Cold War, he predicted that the rest of the mankind history will be calm, there will be no any developments, everything will be stable, and we'll prosper and so on. But we know by now that's definitely not the case. The history evolves, and I'm sure that it's not the advent of AlphaFold, it's it's not the end of the history of structural biology. And the natural intelligence, as suggested by Michael Kashpichnikov, will still dominate. So, of course, a lot depends on the infrastructure of modern research. The science is very technologicized, so to say. Either you have cryo-electron microscopy at your disposal or not. For example, in the United States there are 400 cryo-electron machines. In China there are 40 cryo-electron machines. And that shows the distance you have to close this gap. Either you have a very special modern detector and you see everything at the resolution at 2 angstroms, that means you see everything. Or you have an old detector with a resolution of 4 angstrom or 5 and you see nothing. That's also the difference, and this difference costs several $million probably. So the more you're technologically equipped, the more chances are for your success. And you should look for the teams of the complementary competences. They should be not so single-minded. Probably they should incorporate microbiologists who are very good in putting forward very special problems. Molecular biologists and biochemists who will do all the work for you to prepare your samples, otherwise you're helpless. And you of course need the structural biologists coming from different fields of structural biology, theoreticians who will calculate everything for you with the quantum methods, whatever you ask them, whatever they will calculate for you. Yeah. It's a matter of faith, more or less. And of course, you need some justification. Why do you wasting so many time, so many effort, and so many state money into your research? You need a super task, either a new anti-cancer drug, a new super efficient biocatalyst that will overturn the chemical industry also rather. So you need an idea first, and the rest will come. So that's the end of my story, and that is my teacher, Professor Berezin. He was 19 when I was of your age, when I was a student. He was my director when I was a postdoc, and actually he was my teacher in life and science. This year he is 100 years old, so I would like to dedicate this talk to his memories. And I have 3 affiliations. I'm affiliated to the Federal Center of Biotechnology, where I've worked for nearly 40 years now. I have my alma mater, Moscow State University, which is a co-founder of your university, and Kurchatov Institute in Moscow. And these are the PIs, the project leaders whom I worked with and whose results I presented today. And that is my small lab. Thanks so much.
Thank you very much for such exciting lecture. And now we have the time for the questions. Oh, Professor Shinkarев.
Let me start the discussion. On the structures, what we've shown for enzymes, almost in every cases we see some not aggregation but formation of multimeric assemblies of subunits. Why the nature made these enzymes multimeric? Maybe some allosteric coupling between different active sites like in hemoglobin or not, or active sites are independent.
You say nature is very smart, actually, and it had billions of years to perform each structure, to perform each function. And it depends. For example, if you operate under very unfavorable conditions, for example at high temperature, high salt, you have to hide your surface from the external. You have to multimerize. So, for example, the same enzyme from mesophile or from neutrophile and from alkalophile or semophile will have different oligomeric structure. It will be monomeric for conventional enzyme and multimeric for the extreme operating into extreme condition because it has to hide its internal surface. For example, you need to multiply your subunits if you want to deliver something over a long distance. For example, this is how these multi-heme proteins are multimerized. Yeah? For example, you know about this PD, yes? That is the long protrusions of the protein, protrusions from the cells with electronic conductivity. It has the same conductivity as metal. And with this PD, the cell communicates with other cells and transfers electrons for hundreds of electrons. Yeah? So for each reason, for each function, you have different reasons to multimerize. Sometimes some active sites are combined from two different subunits. When, say, amino acid residues participating in catalysis come from quite two different proteins.
And for butyrylase?
I don't remember actually how it is composed, but probably not. Butyrylcholinesterols, the function of it is not properly understood. In spite of the fact that it is studied for decades. What function which is utilized, which butyrylcholinesterols is interesting to the scientists because it works as an efficient antidote and irreversible bind toxins. Yeah.
Other questions? My question is, what we should do to pave the way for comprehensive understanding about the protein structure, and should we just go the hard way to actually refining our experimental techniques and stimulation calculates, or just based on our previous knowledge to establish a modeling process that can predict protein structure and the function of the protein structure, and which, in other words, what approach we best to choose as an ordinary student, and what is highly productive. Thank you.
If I understood you correctly, you're asking, in short, what is the shortest route to the structure of protein of choice, yeah? It's very difficult to answer. I think the main problem you should first put before you, why I want this structure, what for? Either it's something quite novel, or it's very practical, either it's a target for a potential drug, or it could be the basis of new biocatalysts. Because you need to invest a lot of time, effort, and money to obtain structure. For example, you know that the most precious stones in jewelry are diamonds, yeah? But protein crystals are much more expensive than diamonds. Because if you calculate all the time and money you invest in obtaining one crystal, they will be tremendous sums. So if you want to impress your girl, just present her with a protein crystal, not a diamond. Yeah? That's probably one side of an answer. Yeah? You should need a real problem to tackle. And then there are a lot of routes to go there. And normally, whenever you go to any conference in structural biology, and you will see some professor showing you nature, cell, nature, there's a lot. But he will mention, okay, it took me 10 years to obtain these crystals and nobody wanted, no, everybody failed, but I have this John, he was a student, he was very persistent, and for 5 years he crystallized, and finally we obtained one crystal, and finally we managed to obtain all the data and so forth. So it's a very laborious undertaking with a very few chances for success, actually. It's a long story, yeah? You could be very lucky, of course, with simple things. You can do another protein and that another, it will be 150 structure which is already in the database. It doesn't bring you anywhere. But if you want to do something really novel, you should be so ready for a failure. So there is such a problem. Try, fail, try again, fail, better.
You should be a very brave person to start this work. So, do you have... okay.
I'm just curious about deep sea enzymes. Are there any special data or maybe features of these enzymes? Enzymes, I mean, from maybe animals living at depths like something like, глубоководные, большое давление, как сказывается на работу пропеллера? Вот есть там что-то интересное в этой области?
Честно говоря, я не помню таких работ, да? Но, значит, скорость реакции от давления. I'm sorry for my Russian, I'll send answer in brief in English. Но, по-моему, очень тщательно изучалась, так сказать, скорость реакции ферментативных, в том числе от давления, но там выдающихся каких-то, я что-то не помню, все в соответствии с правилами физической химии, как это и должно быть. So the question was whether there are any data about the enzymes from the deep underwater organism, which are under heavy, so static water pressure. Yeah, I don't know such an examples, but to the best of my knowledge, what I know is that the kinetics of enzyme reactions was studied under high pressures, that meant hundreds of atmospheres of excess pressure, and everything was up to the normal laws of physical chemistry.
Thank you. Any questions? No? Oh.
Thank you for your report. It's very interesting. I have a question about the external fields, and one of the extreme factors of the environment is radiation, and it is well known that some organisms can live in the reactors and in the water of the reactors. Do you know, is it a perspective way to find some new proteins or some new functions in that organisms which can protect from the ionizing radiation? Thank you.
So they specific, they don't live in the reactors, they live near the reactors, yeah? And you see, a protein is just one small piece of the whole organism, yeah? And the whole organism is adjusted to be functional at a certain environment, either high temperature, radiation, or whatsoever. And there are several mechanisms. For example, what is much more better studied is about high temperatures. You could increase the cell stability of the individual protein, or on the other hand, you can increase the stability of the environment of the cell with osmolysis or whatsoever. Yeah? And there are different strategies, and different organisms, they perform different strategies. There is not a universal route. Yeah, and I'm not quite sure that we should look into the individual proteins when we are discussing the problem of the stability of the protein under regulation conditions. So we should look at the whole organism, as the whole thing, how it's all organized, regulated, and so on. Probably just the turnover of the protein is much more rapid. Correction mechanism and so on, replication.
Thank you for your answer. That's all. Thank you again for such interesting discussion, and thank you again, Professor Popov.